Hello everyone, here we are discussing Y.BSc Semester 6, Paper 3, Practical Examination Skeleton Paper. So, in this skeleton paper, we will see the questions one by one. So, question number 1 here you can see is isolation and estimation of RNA by Orsinol method. So, here you don't have to isolate actually. You will be given a standard and unknown solution of RNA, and you are supposed to estimate the concentration of both by Orsinol method. Or there are two methods: either you have to make a series of standard tubes and extrapolate the unknown concentration on the graph, or you are supposed to do with the formula method, in which you are supposed to make three tubes, blank, standard and test, and using the formula you will be calculating the concentration of RNA. The alternative to this question number 1 RNA is isolation and estimation of DNA by diphenylamine method. Same, you can do it by a graph method or formula method. Question number 2 is separation of genomic DNA by agarose gel electrophoresis. Only loading. Here you have only loading for this question. You are not supposed to prepare and cast the gel. You are only supposed to load the gel and answer the questions which are asked by the examiner. How much sample you are loading, what is the staining dye, what is tracking dye, etc, etc, which sample you are loading, like that. So question number 1 carries 15 marks. Question number 2, genomic DNA separation by agarose gel, carries 9 marks. So major question is question number 1, 15 marks. Minor question is agarose gel electrophoresis, 9 marks. The alternative to loading in the agarose gel electrophoresis is colorimetric estimation of proteins by Follin method. So this also you can do by graph method, standard series of graph followed by extrapolation of unknown concentration, or you can also make a 3 tubes and do the formula method and find out a colorimetric estimation of proteins by Follin's method. So you can find out a concentration of a protein. So question number 2 is either electrophoresis or Follin's method, protein concentration using Follin's method. So it is of 9 marks. Question number 3 is problems based on restriction endonucleases. Any two problems you will get. So you are supposed to solve them. The marks are 8 marks. So 4 marks for one question. Data will be given to you, a problem will be given to you, and you have to solve the problem of restriction endonucleases, and it carries 8 marks. Or the alternative to problems is calculation of mitotic index from the photograph which will be given to you. So a photograph which or a stained preparation of onion root tip will be given to you, and you are supposed to calculate a mitotic index. There is a specific formula. Using that formula you are supposed to calculate the number of cells in prophase, metaphase, anaphase and telophase divided by total number of cells. So using this formula you are supposed to calculate a mitotic index. Or the alternative is analyze the given syndrome and comment on the numerical or structural aberration or variation in a chromosome. So a karyotype will be given to you, like Down syndrome 21 trisomy, 18 syndrome, Edward syndrome 18 trisomy, or 13 trisomy, Patau syndrome. So a karyotype will be given to you, and in that karyotype you have to identify. Using that karyotype you are supposed to identify the which syndrome it depicts. Or if it could be a normal also. If there is no syndrome, a normal karyotype can also be given. So you have to identify the syndrome and comment on the numerical aberration or structural aberration. Plus along with this you will get interpretation of genetic formula, deletion, duplication, etc., Robertsonian translocation, etc. So interpretation of genetic formula carries 4 marks, and analysis of syndrome carries 4 marks. So together it will come together for 8 marks. So question number 3 has lots of options. Like the first option is your restriction endonuclease problem, 2 problems for 8 marks. Or you will get calculation of mitotic index, or you will get genetic formula with the syndrome or an karyotype. So there are 3 options in question number 3, and question number 3 carries 8 marks. Question number 4 is demonstrate the use of bioinformatics tool, carries 8 marks. Blast for nucleotide sequence comparison. database at NCBI for querying nucleotide or protein sequence with the help of a suitable operator. So you are supposed to find out a nucleotide sequence and do the blast. PubMed for downloading. So you are supposed to download a research paper using a PubMed with the help of a suitable operator. So NCBI. So question number 4 is related to your bioinformatics, that is carrying 8 marks over here. Now remains your Viva and journal. Journal carries 5 marks and Viva carries 5 marks. Viva could be a table Viva, or it could be based on a theory, or it could be based on a practical component. So question 1, DNA or RNA, 15 marks. Question 2, agarose gel electrophoresis, only loading, 9 marks. Or Follin protein estimation by Follin method, 9 marks. Question number 3, restriction endo problems or mitotic index, or it could be analysis of syndrome and interpretation of genetic formula. Question number 4, bioinformatics. Question number 5, Viva, and question number 6, journal. Thank you so much. All the best for your exam.